Okay, so this is chapter 5, part 2. We'll finish up the chapter. We've got to get through active transport and bulk transport, and then we'll be done with chapter 5. So we've already discussed passive transport, diffusion, osmosis, facilitated transport. All of those were passive processes, which means that ATP was not required, just a gradient, a concentration gradient across the membrane. Okay? Now, active transport is the complete opposite. So active transport is going to require ATP energy. And now we're going to move substances up their concentration gradient or against their concentration gradient. So this is how cells will move substances from where they're at a high concentration, from where they're at a low concentration to where they're at a high concentration. So literally pumping molecules from one side of the membrane against their concentration gradient. We're going to need energy to do this. Okay? I want to introduce you to a term called electrochemical gradient. In all living cells, there's going to be a charge difference across the plasma membrane, and that charge difference has to be created or generated by the cell. Okay? And how do those cells do it? They're going to build up concentrations of different, charged particles, which we call ions. Okay, and the figure on the next slide is going to show this. When we have this electrochemical gradient established, what ends up happening is the interior, the cytoplasmic side of the membrane ends up being negative or overall negative in charge, and the extracellular side of the membrane, the outside, has an overall positive charge. Okay? There is one particular active transporter called the sodium-potassium pump that is the main player in establishing this electrochemical gradient, this charge difference across the membrane. And so it's created mainly by the distribution of sodium, which is Na+, and potassium, which is K+, ions across that membrane. Okay? So let's take a look. So first off, here is our phospholipid bilayer, right here. This is the inside of the cell, the cytoplasmic side of the cell, and this is the extracellular fluid. Okay, and right here is a membrane transporter that's transporting some stuff across the membrane. The electrochemical gradient, again, is going to be the combined effects of a concentration gradient, so a difference in the concentration of, in this case, ions across the membrane, and an electrical gradient. Okay, so if we look here, there's definitely a concentration gradient for everything here. For sodium, which is Na+, right? There's a lot more sodium outside the cell than in. Let's look at potassium, K+. Lots of potassium inside the cell, very little outside. We could say the same thing for, the chloride ions. So lots of chloride ions outside the cell, very few inside. Okay, and so we can see that there is definitely a concentration gradient. Let's look at the electrical gradient. There is definitely a lot more positively charged ions on the outside than on the inside of the cell. So again, the inside, the cytoplasmic side of the membrane, tends to, in most cells, have an overall negative charge, where the extracellular fluid, the outside of the cell, has an overall positive charge. And this electrochemical gradient is established by active transporters. Okay, so let's talk about these active transporters. So again, active transport, the proteins involved here, we're going to call them pumps. They are literally pushing substances against their concentration gradient. Okay. The job of an active transporter is to maintain concentrations of ions and other molecules across that membrane in the face of passive transport. So if I go back a slide, this protein right here is going to be a facilitated transporter that is responsible for passively moving potassium ions across that membrane. Notice how it's going to grab this potassium ion and move it right across. So passive transport is constantly happening, And so active transport has to be constantly keeping up with it, trying to maintain those electrochemical gradients. Okay? A huge amount of energy, a huge amount of ATP is going to be spent trying to establish these electrochemical gradients. Okay? Now there's two types of active transport. Primary and secondary, nicely named for us. Primary active transport, these are going to be pumps that are going to move ions across the membrane to create that electrochemical difference, that charge difference across the membrane. ATP will be used directly. So that energy source that we call ATP, it's going to be used directly. The pump is going to be activated by ATP. The pump will use the energy supplied by ATP to move stuff across the membrane. Now, once this charge difference, once this electrochemical gradient is established by these primary active transporters, then we get secondary active transport. And so secondary active transport is the movement of materials against their concentration gradient by using the established electrochemical gradient. So ATP is not used directly. Instead, the electrochemical gradient produced here is used. Let me show you an example. Oh, wait, before we get there, let me show you these. So all of the proteins involved in active transport, whether it's primary active transport or secondary active transport, they're all going to fall into one of these three categories. So we have pumps that are uniports. A uniporter protein is going to carry one specific ion or molecule across the membrane. A symporter will carry two different ions in the same direction across the membrane. And the last type, which the sodium-potassium pump, which we're going to talk about in a second, falls into this category, is the antiporter. The antiporter carries two different ions or molecules in opposite directions. So one molecule is brought into the cell while the other is brought out of the cell. Okay? So those are the three types of proteins. Again, all are carriers. Remember back to the previous video, facilitated transport? We said that proteins involved there could be carriers or channels. In this case, with active transport, there's no channels. All proteins involved here are carriers. Okay, so let's look at an example. Here's an example of primary active transport. So this is going to be a carrier protein in the membrane that's going to transport materials against their concentration gradient with the use of ATP. In this particular example, this is the sodium-potassium pump. This is the textbook example that's always used for active transport. It's called an electrogenic pump because it sets up an electrochemical gradient, a charge difference across that membrane. Okay, it's going to transport sodium, which is shown here in as orange hexagons, and potassium, which is shown here as yellow ovals. Okay, now this is going to be an antiporter, meaning it's going to transport the two types of cargo in opposite directions. So this is going to bring sodium out, and it's going to bring potassium into the cell. Okay, so let's set this up. Here's the plasma membrane. Here is the plasma membrane. Here is outside of the cell, the extracellular space, and here's the cytoplasm. Okay, so let's look at the concentration first of these ions. So look inside the cell at the cytoplasm. There is a low concentration of sodium. So if you look here, you can see that there's a low concentration of sodium, okay, inside the cell. Let's look outside the cell. There's a high concentration of sodium outside the cell. Okay, so let's first follow the movement of sodium. Sodium is going to, so we've got sodium here and here. It's going to bind to our carrier protein on the cytoplasmic side of the cell. ATP, energy, is going to come in, and that's going to cause this carrier protein to change shape and to dump off the sodium on the extracellular side of the cell. So what just happened? We just moved 3 sodium ions from low to high against their concentration gradient. You see that? So we just pumped 3 sodium ions from where they were at a low concentration to where they're at a high concentration. So we just went against their concentration gradient. We pumped those ions out. That required ATP. Okay? Again, that protein is a carrier protein. It changed shape in order to deliver that sodium to the outside of the cell. Okay, now what happens next? Well, now potassium is going to be moved. So let's look at the concentration gradient of potassium. So look outside the cell. I don't see much potassium. So if we look at potassium, K+, outside the cell there is a low concentration. And if we look inside the cell, there's a high concentration. Well, guess what? We're also going to pump potassium against its concentration gradient from low to high. Now in this case, we're only going to pump 2 potassium in. Okay, that's how the sodium potassium pump works. It pumps 3 sodium out and 2 potassium in. Okay, so the potassium binds here. That ATP comes off as adenosine diphosphate. Okay, we'll talk about ATP a little bit more in some coming up chapters. Okay, that causes, again, that protein to change shape and to dump off the potassium inside of the cell. So we pumped out 3 sodium and we pumped in 2 potassium. Okay? So we're just increasing the gradient here, right? We're adding more sodium outside the cell, making the concentration gradient of sodium even larger. And the same thing with potassium. By pumping potassium in, we just brought in more potassium, making the concentration gradient across that membrane even greater. Let's think about charge. We pumped 3 positively charged ions out and only brought 2 positively charged ions back in. So what's going to happen is the outside of this membrane is going to become more positively charged compared to the inside, which is going to have less positive charge, or it's going to be more negative. Okay, so not only are we setting up a concentration gradient of these ions, but we're also setting up an electrical gradient. So we're setting up an electrochemical gradient across the membrane. Okay, so here's the key textbook example, the sodium-potassium pump. Again, energy is going to be used in the form of ATP to pump out 3 positively charged sodium and bring in only 2 positively charged potassium. We're setting up an electrochemical gradient. Again, the proteins involved here are carrier proteins. This is an example of primary active transport because ATP was used. What did it do? It provided the energy for that carrier protein to change shape. Okay. So, let's look at secondary active transport. So we just set up a concentration gradient of both sodium and potassium, right? So let's use what we just set up. So these are sodium ions. Okay, so let's focus on those first. So if we look at the concentration of sodium outside the cell, it's high, and inside the cell, it's low. So based on the basic laws of diffusion, right, or facilitated transport, sodium should move into the cell. It should move down its concentration gradient. Okay, so what that represents here is a form of potential energy. We have a gradient. Okay, let's look at glucose. In this particular example, this is the sodium glucose symporter. So first of all, it's a symporter. So it's going to be moving 2 different types of cargo in the same direction. Okay, what's its cargo? Sodium, Na+, and glucose. Glucose is represented here by these rectangular orange pieces. Let's look at the concentration of glucose. Well, inside the cell, there's a high concentration of glucose, and outside the cell, the concentration is pretty low. So it's going to take some energy to move glucose into the cell from low to high, right, against its concentration gradient. So where's that energy going to come from? It's going to come from the sodium concentration gradient. So how the glucose sodium symporter works, that's shown right here, this guy. In order to transport either sodium or glucose across the membrane, both have to bind. So sodium wants to move down its concentration gradient, so it will bind here to sodium ions. And the glucose is going to bind as well. And the movement of sodium down its concentration gradient is going to provide the energy for this protein to change shape. And so just by sodium and glucose binding, that's going to cause a change in shape of that protein, and the cargo will get dumped off on this side of the membrane. So the sodium moved down its concentration gradient, but the glucose moved up its concentration gradient. How? What provided the energy? The sodium concentration gradient. Okay, so again, that's an example of secondary active transport, or co-transport is another name for this. ATP wasn't used directly. Instead, the energy came from the gradient, the sodium gradient. Okay, that was that source of potential energy. Okay, so that's how glucose is oftentimes moved into cells. Okay, so take a pause here. Make sure you understand what active transport is, and make sure you understand the difference between primary and secondary active transport. Is ATP used directly? Yes, in primary. In secondary active transport, no. Okay, so make sure you describe the differences there. When you're finished, come back to me. I got some questions. Okay, so the first question. Active transport must function continuously because red, the plasma membranes wear out. Blue, not all membranes are amphiphilic. Green, osmosis dilutes the solute concentration inside the cell. Or yellow, diffusion is constantly moving solutes in the opposite direction. I'll give you a minute to answer that. So yep, yellow. So because diffusion is constantly moving solutes in the opposite direction, and your cells need to maintain that electrochemical gradient, active transport has to continuously be active. Next question. How does the sodium-potassium pump make the interior of the cell negatively charged? Is it by expelling anions, by pulling in anions, by expelling more cations than are taken in, or yellow, by taking in and expelling an equal number of cations? So remember, an anion is a negatively charged ion, and a cation is a positively charged ion. So the sodium-potassium pump in particular. So what does it do? The sodium-potassium pump, remember, it's going to pump out 3 positively charged sodiums and pump in only 2 positively charged potassiums. And so green, the pump is going to push out or expel more cations than are taken in. Okay, next question. What is the combination of an electrical gradient and a concentration gradient called? Is it called a potential gradient, an electrical potential, a concentration potential, or an electrochemical gradient? Hopefully you answered yellow. It's an electrochemical gradient. Next question. In secondary active transport, red, ATP is used to drive the movement of a molecule against their concentration gradient. Blue, uniporters are always used. Green, the electrochemical gradient of one molecule is used to drive the uptake of a second molecule. Or yellow, both molecules are moved down their concentration gradients. And hopefully you answered green, right? So the electrochemical gradient in our example was that sodium gradient. It's going to be used to drive the uptake of, in our case, it was glucose, that second molecule. Okay, we're real close to being done here. So the last part of the chapter, transport. So instead of just transporting 1 or 2 molecules, what if we want to transport a lot of molecules all at the same time, or a large amount of fluid, or large things like whole bacterial cells? How are we going to move that kind of stuff across a membrane? That's going to be brought in or brought out via bulk transport. Okay, so moving large particles or large amounts of substances. Bulk transport requires energy, so it is a form of active transport. ATP will be burnt or used in this process. We've got a couple of types. So endocytosis, when you see that endo, that means to internalize, so to take into the cell, moving particles or fluid into a cell. I'm going to show you some pictures of this, but essentially what happens is the plasma membrane is going to fold in, or we use the term invaginate, and it's going to form a pocket around whatever is being brought in. The pocket will eventually pinch off, and the particle or particulate matter will end up in a newly formed vesicle formed from the plasma membrane that will now be in the cytoplasm. Again, I'll show you a picture of this. There's 3 types of endocytosis. There's phagocytosis, which I like to call cellular eating. It's the uptake of particles, particulate matter, in a non-specific way. Penocytosis, I like to think of as cellular drinking. It's the uptake of large amounts of solvent. Okay, and sometimes there's solute in there as well. And the last type is receptor-mediated endocytosis. Okay, this is the same as phagocytosis, so taking up particulate matter, except for now it's specific in that there has to be molecular recognition. So whatever's coming in has to first bind to a receptor to be taken inside. And again, I'm going to show you all of these. Okay, so let's look at the first one, phagocytosis. This is a form of endocytosis. It requires energy, and we've got stuff being taken into the cell. So here is the plasma membrane. This area here is the cytoplasm, and this is outside of the cell. So we have a large particle. Maybe it looks like a bacteria or something like that. And the cell wants to take that in. And so what's going to happen is the plasma membrane is going to form a pocket, or it's going to invaginate. The particle goes into that pocket. Eventually, this is going to pinch off. The membrane will just seal right up. And now we have a membrane-bound vacuole that has our particle in it. Okay, so that particle has been taken up. It's been phagocytosed. Okay. Okay. Penocytosis is really the same thing, right? Taking up stuff into the cell. It's a form of endocytosis that does require energy. Instead of taking up these large particles, now we're taking up fluid that might have some solute in it. Okay, so again, this invagination or pocket forms, the fluid's going to move into there. Eventually that's going to pinch right off, and we end up with an intercellular vesicle that has that fluid in it. Okay. The next one is receptor-mediated endocytosis. Again, requires energy. This is when particulate matter, here it's the stars, I'm not sure what it is, it needs to get into the cell, but it's going to bind specifically to a receptor first before it gets internalized in a membrane-bound compartment. Okay, so bulk transport of stuff into the cell is called endocytosis. We have phagocytosis, we have penocytosis, and we have receptor-mediated endocytosis. All 3 are going to bring materials into the cell and require energy. Okay, so how about when stuff needs to leave? Okay, like if you're secreting hormones from some cells, that's going to be called exocytosis. So the cell is going to move materials in bulk, wants to push a bunch of stuff out all at once. What's going to happen? So in our cytoplasm, we'll take whatever needs to be secreted, whatever needs to be released. We'll surround it by a membrane. That vesicle will move toward the membrane. It'll fuse with it, and then its contents will be dumped to the extracellular fluid, dumped out of the cell. So again, your cell might do this to get rid of waste products in bulk, or if it's a cell involved in secretion of things like hormones, it will employ this method, again called exocytosis. Requires energy. It's the bulk transport of materials out of the cell. Okay, so this is our last Are You With Me? I want you to compare and contrast the types of bulk transport. Sketches here would be great too. I didn't really ask for them, but if you want to provide them and then provide explanations for each so that I know that you understand them, that would be great. When you're finished, come back. I've got a few more questions. Okay, so the first question. What happens to the membrane of vesicles after exocytosis? Do the membranes leave the cell? Are they disassembled by the cell? Do they fuse with and become part of the plasma membrane? Or are they used again in another exocytosis event? So let's back up here. So again, the vesicle moves to the membrane. Ooh, there's a fusion, and then everything gets dumped. So notice this part, this part right here, this is new membrane from that vesicle that's now fused with the plasma membrane. So your answer here would be green. It fuses with and becomes part of the plasma membrane. Next question, which transport mechanism can bring whole cells into a cell? Pinocytosis, phagocytosis, facilitated transport, or primary active transport? Hopefully you answered phagocytosis, right? That's going to be bulk transport of large things like a cell into a cell. Okay. In what important way does receptor-mediated endocytosis differ from phagocytosis? Red, it transports only small amounts of fluid. Blue, it does not involve the pinching off of the membrane. Green, it brings in only specifically targeted substances. Yellow, it brings substances into the cell, while phagocytosis removes substances. So hopefully you picked green. So receptor mediated is going to be only specific substances are going to be brought into the cell, where phagocytosis, there's no binding of anything to a receptor, so it's a non-specific uptake. Next question. Imagine a cell can perform exocytosis, so it can release stuff out, right? The membrane of that vesicle would fuse with the plasma membrane, just fine. But that cell could only minimally endocytose, so it really can't take stuff in so well. What would happen to the cell over time? The cell would secrete all of its intracellular proteins. The plasma membrane would increase in size over time. The cell would stop expressing integral receptor proteins in its plasma membrane. Or yellow, the cell would lyse. It's a tricky one. Hopefully you answered blue. So exocytosis is happening, which adds to the size of the membrane, where endocytosis is not happening so much, right? So endocytosis would decrease the size of the membrane because the membrane is being taken in. So if you have exocytosis happening but not endocytosis, then the size of that plasma membrane would increase over time. Okay, that's the last slide. So you want to make sure that you have all of your Are You With Me's done. Remember, if it asks you to sketch, make sure that you're sketching. And you want to go ahead and you want to upload your Are You With Me in-class assessment right to Blackboard. Okay. Once you're finished with this, you want to go ahead and study all of your notes that you've taken so far, and then you can go ahead and you can take the quiz for Chapter 5.